Hello everyone, I'm Dr. Sonia Angelin, Associate Professor, Department of Life Sciences, Krista Jayanti College. In this video, I will be discussing about replicative and non-replicative transposition. There are several methods of genetic recombination in organisms, and transposition is a specific form of genetic recombination which moves certain mobile genetic elements called transposable elements, which are addressed as transposons. Movement occurs through recombination between the DNA sequence at the very end of the transposable elements and a sequence of DNA of the host cell. This can happen in two different ways, by replicative method and by non-replicative method. Transposons are transposable elements also known as jumping genes, and they are the examples of mobile genetic elements. They were first identified by Barbara McClintock in the year 1983. A transposon is a DNA sequence that can insert itself or copy of itself in a new location in the genome without having any sequence relationship with the target locus. Replicative transposition is a type where the element's DNA is duplicated during each transposition. The first step of replicative transposition is the assembly of the transposase protein on the two ends of the transposon to generate a transposome. The next step is DNA cleavage at the ends of the transposon DNA. The transposase enzyme introduces a nick into the DNA at each of the two junctions between the transposon sequence and the host DNA. The intermediate generated by DNA strand is a doubly branched DNA in which the 3' ends are joined to the new target site and the 5' end remains joined to the old flanking DNA. The two branches of this intermediate has structures of a replication fork. So this replication reaction generates two copies of transposon DNA. This is a pictorial representation showing you that from the donor, a copy of it is being inserted into the recipient DNA. This is a replicative transposition. Non-replicative transposition is the transforming element moves as a physical entity directly from one site to another, and it is conserved. The transposon will leave from its original position and insert into a new position in the receptor. In this mechanism also, transposome formation occurs. After that, the transposase enzyme brings the two ends of the transposon DNA together to generate a stable DNA protein complex called transposome. The next step is excision of the transposon DNA from its original location, followed by the 3' OH end of this DNA attacking the site of the new insertion. As a result of this, the transposon DNA is covalently joined to the DNA at the target site. The transposome ensures that the two ends of the transposon DNA attacks the two DNA strands of the same target site together. After non-replicative transposition, the donor may shorten while the receptor may lengthen. This is a pictorial representation showing you the non-replicative transposition. As you can see, the transposon is being excised with the help of the transposase enzyme, and it is being inserted into the target site, and which results in a successful transposition. These are the references. Thank you.